The central premise of synthetic biology is the idea that we could program living cells much like we program computers. The tools for this are what we call synthetic gene circuits, which function as small biological programs. However, biology is inherently complex and often unpredictable. In this explainer, we'll examine this challenge and explore how artificial intelligence is being introduced as a potential solution.
So here's how we'll break it down. We'll start with the fundamentals of programming biology. Then, we'll look at the main obstacle in the field, something known as the context problem. After that, we'll see how machine learning presents a new, data-driven solution. We'll also examine a hybrid approach that aims to combine the best of both worlds. And finally, we'll touch on the significant challenges that still lie ahead.
All right, so let's start with the basics. How does one actually go about programming biology? The objective is to engineer living systems in a predictable way, and the primary tool for this is the synthetic gene circuit.
You can think of a synthetic gene circuit as a kind of biological logic gate for a cell. It's constructed from components like DNA and proteins and is designed to do something specific. It senses an input signal, say the presence of a certain molecule, and then it triggers a predefined response, like producing a therapeutic protein or activating a fluorescent marker. The development of these circuits follows a classic engineering cycle: design, build, test, and learn. First, you conceptualize the function. Then you build the required DNA. After that, you test it inside a living cell to see what happens. And finally, you learn from the results to refine the next design. This is an iterative process. But, as we're about to see, the test and learn steps in biology can be far from straightforward.
This brings us to what is perhaps the single greatest challenge in synthetic biology. That clean, neat engineering cycle we just mentioned often breaks down. And the reason is that the internal environment of a cell is a highly complex and dynamic place, which makes engineering life quite difficult.
Here's the core issue. Biological components that work as expected in isolation often behave differently when you assemble them together. An engineer can design a circuit using well-understood parts, but when that circuit is placed inside a cell, its performance can deviate significantly from the prediction. This suggests there are hidden interactions that the initial design did not account for.
This phenomenon is known as context dependence. The behavior of a biological part is not fixed. It can change depending on its surroundings. Everything from its position on the chromosome to the overall metabolic state of the cell can alter its function in ways that are difficult to predict.
So, what causes this? Well, there are several factors. One is resource sharing. The cell only has a finite amount of machinery, like ribosomes and polymerases. Your engineered circuit has to compete for these resources, which can affect its performance. Another factor is retroactivity. This is where adding a component to the end of a circuit can feed back and unexpectedly alter the behavior of components at the start. And finally, there are simply unforeseen interactions between the engineered circuit and the host cell's native machinery.
To address these limitations of traditional, physics-based modeling, researchers are now turning to machine learning. This marks a shift toward a more data-driven approach for both understanding and engineering these biological systems. Now, while traditional biophysical models try to predict outcomes based on established physical laws, machine learning models take a different approach. They are designed to learn complex patterns and relationships directly from large sets of experimental data. This means they have the potential to capture those subtle, hidden interactions responsible for context dependence, even if the underlying biological mechanisms aren't fully understood. And researchers are employing a wide variety of machine learning techniques. There are different types of neural networks for prediction, reinforcement learning to optimize designs, active learning to make experimentation more efficient, and even generative models that can design completely new biological sequences. The diversity of these tools reflects the complexity of the problem. The application of machine learning here can be separated into two broad predictive tasks. The first is sequence-to-function prediction. The model is given a biological sequence, like DNA or a protein, and its goal is to predict the function of that single component. This is a more established area. The second and more difficult task is composition-to-function prediction. Here, the input is a whole collection of genetic parts, and the goal is to predict the behavior of the complete assembled circuit. This remains a grand challenge for the field.
Recognizing the limitations of relying exclusively on either mechanistic models or pure machine learning, a new direction has emerged. This is the development of hybrid approaches that try to combine the strengths of both paradigms.
Let's just compare the two approaches directly. Traditional, mechanistic models are interpretable. We understand their internal workings, which means we can use them to guide design changes.
Their weakness is that they often fail to capture the full complexity of biology. Machine learning models, on the other hand, have high predictive power for complex systems, but they typically require very large datasets and can struggle to generalize to conditions they haven't been trained on.
So the idea behind a hybrid model is to let each method do what it does best. You start with a traditional physics-based model to provide a baseline prediction. Then, you use a machine learning model to learn and correct for the errors of that initial model. The ML component essentially captures all the complex biological effects that the first model missed, which makes the combined prediction more accurate.
This hybrid approach offers several key advantages. You retain the interpretability of the mechanistic model while gaining the predictive accuracy of machine learning. It's also hypothesized that these models may perform better when faced with new, out-of-distribution data. And, importantly, because the machine learning component is only correcting errors rather than learning from scratch, these models can often be trained with smaller datasets, which is a significant practical benefit.
Now, despite the clear potential of both machine learning and these hybrid models, there are several major challenges that need to be addressed before these approaches can have their full impact on synthetic biology.
But getting there is not simple. First, there's a need for deeply interdisciplinary teams, where experts in machine learning, biology, and modeling can collaborate effectively. There's also the practical difficulty and high cost of generating the large, high-quality datasets that these models require. On a technical level, even when individual parts are designed using ML, composing them together can still lead to errors. And perhaps most critically, there's currently a lack of theoretical guarantees on the functionality and safety of these AI-designed circuits.
This issue of safety and reliability is a major roadblock. If the goal is to use engineered cells for therapeutic purposes inside the human body, or to release them into the environment for bioremediation, we need a very high degree of certainty about their behavior. The black box nature of some machine learning models makes it difficult to provide that kind of formal guarantee at this time.
So, in conclusion, the merger of machine learning and synthetic biology offers a promising path forward. It provides new tools to manage the complexity that has historically made engineering biology so challenging. Hybrid models in particular appear to be a valuable approach. However, substantial challenges related to data generation, expertise, and most importantly, safety still remain. This leaves the field with a central question for its future. As we continue to merge artificial intelligence with biological code, how do we ensure that the resulting designs are not only powerful, but also predictable and safe?